I am Khushi from chemistry department. Today we were discussing about enzyme inhibitors and their importance and translations. What is translation? Enzymes are soluble colloidal organic catalysts formed by living cells, specific in actions, protein in nature, inactive at 0 degrees Celsius and destroyed by most heat at 100 degrees Celsius. How enzyme catalyze reactions? Enzymes brings reactants together and position them correctly so that they easily attain their transition state configuration. Enzyme weakens bonds in the reactants. Enzymes may participate in the reaction mechanism. Enzymes form strongest interactions with the transition state than with the substrate or the product. Basically, enzyme provides a alternative path for a reaction by lowering its activation energy so that reaction completes easily and fastly. Now we are discussing about what are inhibitors. Inhibitors are the chemicals that reduce the rate of enzymatic reactions. They are usually specific at work at low concentrations. They block the enzyme but they do not usually destroy it. Many drugs and poisons are inhibitors of enzymes in the nervous system. Enzyme inhibitors. Enzymes inhibitors are the molecules that reduce the catalytic activity of enzymes, reducing the effective activity or complete blocking of enzymes may cause either complete death of cells, either modification in the pathways. Here we are discussing about types of enzyme inhibitors. There are three types of enzyme inhibitors. First one is reversible, second one is irreversible, third one is allostatic. In reversible, there are three types: competitive, non-competitive, and competitive. In irreversible, there is suicide inhibitor. Reversible inhibition. Inhibitor binds to the enzyme reversibly through a weak non-covalent interaction. An equilibrium is established between the free inhibitor and the EI complex and is defined by a equilibrium constant Ki. Reversible inhibitors depending on the concentration of E, S and I, where E is enzyme, S is substrate and I is inhibitor, show a definite degree of inhibition which is reached fairly rapidly and remains constant when the initial velocity studies are carried out. Competitive inhibition. The inhibitor which closely resembles the real substrate is regarded as a substrate analog. The inhibitor completes with substrate and binds at the active site of the enzyme but does not undergo any catalysis. As long as the competitive inhibitors held the active site, the enzyme is not available for the substrate to bind. During the reaction, ES and EI complex are formed. The relative concentration of the substrate and inhibitors and their respective affinity with the enzyme determines the degree of competitive inhibition. The inhibition could be overcome by a high substrate concentrator. In competitive inhibition, the Km value increases, where Vmax value remains unchanged. The diagram for the competitive inhibition. Non-competitive inhibition. The inhibitor binds at a site other than the active site on the enzyme surface. The binding impairs the enzyme function. The inhibitor has no structural resemblance with the substrate. However, there usually exists a strong affinity for the inhibitor to bind at the second site. In fact, the inhibitor does not interfere with the enzyme substrate binding, but the catalysis is prevented, possibly due to distortion in the enzyme conformation. The inhibitor generally binds with the enzymes as well as EX complex. For non-competitive inhibition, the Km value is unchanged while Vmx value is lowered. Heavy metal ions can non-competitively inhibit with enzymes by binding with groups. Uncompetitive inhibitions diagram. Here we can say that there is an equilibrium condition Irreversible inhibition. Inhibitors binds at or near the active site of the enzyme irreversibly, usually by covalent bonds, so it cannot dissociate from the enzyme. No equilibrium exists. Effectiveness of I is expressed not by equilibrium constant but by a velocity constant determines the fraction of the enzyme inhibited is given period of time by a certain concentration of the AI. Now, as we know that protein is formed from RNA, when DNA forms another DNA replicates itself, and when RNA formed from DNA, then this is called transcription, and when RNA form protein, that is called translation. Transcription carried by RNA polymerase. Translation is the process on ribosomes. Replication is carried out by the DNA polymerase. Translation forming of a polypeptide uses mRNA as a template for AA sequence. 4 steps. It involves initiation, elongation, translocation, and termination. Begins after mRNA enters cytoplasm. Uses tRNA as the interpreter of RNA. Lysosomes made of proteins and RNA. Each has large and small subunit. Each has 3 binding sites of tRNA on its surface. Each has 1 binding site of mRNA. Facilitates codons and anticodons bonding. Translation. The 3 tRNA binding sites are A site, P site, and E site. A site holds tRNA that is carrying the next amino acid to be added. P site holds tRNA that is carrying the growing polypeptide chain. Polypeptide chain. E sites where discharged tRNAs leave the ribosomes. Here's the structure where we can see that larger subunit, exit sites, mRNA, small subunits. The genetic code. 4 RNA nucleotides are arranged 20 different ways to make 20 different amino acid. Nucleotides bases exist in triplets. Triplets of bases are the smallest unit that code for an AA. 3 bases is equals to 1 codon, which is equals to 1 AA. There are 64 possible codons. 64 is equals to 4Q. The genetic code. Most of the 20 AA have between 2 and 4 possible codes. The mRNA based triplets are codons. In translation, the codons are decoded into amino acid that make a polypeptide chain. 61 or 64 codons code for AA. Codons. AUG has 2 functions. Code for amino acid methionine functions as a start codon. mRNA codon AUG start translation. The 3 uncounted for codon act as stop codons. How it works: DNA antisense ACC AAA CCG, mRNA transcription UGG UU GGC, polypeptide translation tryptophan, phenylalanine, and glycine. Now we are discussing about the process of translation in brief. First one is initiation. Bring together mRNA and tRNA and ribosomal subunits. Small ribosomal subunit binds to mRNA and inhibitor tRNA. Start codon is AUG. Start anticodon is UAC. Small ribosomal subunits attached to 5' end of mRNA. Downstreams from the 5' end is the start codon AUG. The anticodon UAC carries the AA methionine. After the union of mRNA, tRNA and small subunit, a large ribosomal subunit attaches. Here initiation is complete. The inhibited tRNA and AA will sit in the P site of the large unit. The A site will remain vacant and ready for the amino acid tRNA. Translation elongation. This is the second step. Amino acids are added one by one to the first amino acid. Remember, the goal is to form a polynucleotide. Codon recognition. mRNA codon in the A site forms hydrogen bond with the tRNA anticodon. Peptide bond formation. The tRNA will the polypeptide chain in the A site is translocated to the P site. This one is the third step. tRNA at the P site moves to the E site and leaves the ribosomes. The ribosomes moves down the mRNA in the 5' to 3' and the direction is remain constant 5 dash to 3 dash. Last step is termination happen at the stop codon. Stop codons are UAA, UAG and UGA. They don't code for AA. The polypeptide is freed from the rhizomes and the rest of the translation assembles comes apart. Thank you.